So what I'm going to talk to you today is very friendly manner, the nanotechnology in controlling this pandemic, COVID-19. So the contents go as follows. I'm going to briefly introduce what is COVID-19 and then about SARS-CoV-2 virus, then infection mechanism and lifestyle of SARS-CoV-2 in the host cells. Then I'll show you SCM and SCM, scanning electron microscopic and transmit and electron microscopic images of cells infected with SARS-CoV-2. And then we talk about SARS-CoV-2 variants and evidence for reduced antibody protection against SARS-CoV-2 variants. And then we talk about different types of vaccines, risk associated with fast-track vaccine evaluation, as I take from standard journals like Nature, and then ultra-potent COVID-19 vaccine designed via computer is based on some nanotechnology, and nano insights into diagnosis, treatment, and prevention of COVID-19. Then metal and metal oxide nanoparticles in COVID-19 control. And finally, I will show you our R&D activities and what we have done in our capacity to actually to control getting this infection. So what is COVID-19? It is a severe acute respiratory syndrome caused by the coronavirus called SARS-CoV-2. The disease caused by this virus is called COVID-19, as we all know. Since the first description of the coronavirus-related pneumonia outbreak in Wuhan province of China in December 2019, the virus SARS-CoV-2 that causes the infection or the disease, COVID-19, has evolved into a pandemic as we know now. Globally, as of 15th October 2020, yesterday, there have been 239.4 million confirmed cases of COVID-19, including more than 4.8 million deaths reported to WHO. As of 13 October 2021, a total of nearly 6.5, more than 6.5 billion vaccine doses have been administered. That is where I got it from, covid19.who.in. You can go to that and verify these data. Now you see these things in the graphical mode in different countries, America in this color, and Europe, Southeast Asia, Eastern Mediterranean, Western Pacific, Africa, etc. And you can clearly see there are three, we have three maxima in these curves, showing that three stages in this COVID-19 pandemic. That is clearly seen if you take the total cases. You see in this year, actually there are three maxima in these COVID-19 waves. So this is the virus. That is this schematic structure of SARS-CoV-2 virus. The viral structure is primarily formed by the structural proteins such as spike S. So you have S1 and S2, the spikes. These things are these are the spike proteins, and then membrane M. This is a lipid membrane M, and envelope E. This is the envelope E, and nucleocapsid N proteins. So these are the kind of proteins by which this SARS-CoV-2 virus is made of. The S, M, and E proteins are all embedded in the viral membrane. As you see, all these proteins are embedded in the viral membrane. The N protein interacts with the viral RNA. This is an RNA virus, RNA into a core of the virion. So this is how say N protein, the N protein nucleocapsid. This one interacts with that RNA. So this is a virus. The virus is not a live species. It becomes live only when it goes into a host cell. So I'm not going to go into details of this infection mechanism and lifestyle of SARS-CoV-2 in the host cell, but briefly, SARS-CoV-2, then it binds to the spike proteins to your particularly ACE2 receptors in your nose, mouth, etc. They are more expressed in nose, and then it releases a viral genome and ribosome, and then translation of viral polymerase goes into RNA replication and transcription, and eventually to get you infected with the virus. And these are some SCM images. As you see, this is the novel coronavirus SARS-CoV-2. This scanning electron microscopic image, when you zoom this virus is like, their virus is like 125 nanometers. When you magnify it using electron microscopy, you can see them. SARS-CoV-2 round, these are round gold objects, are SARS-CoV-2 viruses emerging from the surface of cells cultured in the lab. So you see large number of viruses emerging from the cells cultured in the lab. The virus shown is isolated from a patient in the US. I have taken this from Wikipedia, so it's from a US patient, so it's freely available. I can take them. And basically, when it is infected, the colorized scanning electron micrograph of apoptotic cell, red cell, and in red, heavily infected with SARS-CoV-2. You can see these very small yellow dots are these SARS-CoV-2 viruses. So these cells are heavily infected with virus. So many millions, trillions of viral particles on the cells. And again, and different types of samples infected with SARS-CoV-2. You can see that SARS-CoV-2, it infects in large amounts. And when you can see scanning, you scan the surface of the material. But if you use the transmission mode of the electron microscopy, the electrons go through the sample. You have to have very thin layer. So viruses are very thin. So you can see the structure of the virus. So you see this is transmission electron microscope of SARS-CoV-2 viral particles isolated from a patient. You see these particles, so many particles are there. So having understood what SARS-CoV-2 virus is that is causing this COVID-19, now we know that this virus goes on making different variants. So the original SARS-CoV-2 virus has been subjected to various mutations to result in the evolution of strong and more infective variants. As you know, now Delta, Epsilon variants are more infective and very strong, threatening to the human lives. The currently spreading SARS-CoV-2 variants among the global population include 4 dominant ones, as you know, which are we usually call alpha, beta, like that. For simplicity, alpha variant is formerly called UK variant, or officially referred to as B.1.1.7, B117, first found in London and Kent in UK. And then beta variant, formerly called the South African variant, and officially referred to as B1351. The gamma variant, formerly called Brazil variant, and officially referred to as P1. And the Delta variant, formerly called the Indian variant and officially referred to as B1617.2. So you see that, I mean, we have this real nasty situation because of this Delta variant these days. And luckily, we are now controlling the situation, but still, I mean, in the first place, our total deaths were limited to 30. Now it's really high because of this highly infective and strong variants coming. Additionally, there are various other variants such as L452R, E484K. Likewise, there are various other variants increasing the danger of current COVID-19 pandemic. You can see various variants. See different UK variant, and then South Africa variant, and Brazil variant, and Indian variant. They are the most prominent and dominant variants of this SARS-CoV-2 virus. See different variants like antibody-mediated protection after convalescence is high, and this variant reduce. Antibody-mediated protection after vaccination is high for the original virus, and the UK version is high, and it's reduced there. So likewise, when you get more and more dangerous viruses, the protection becomes less and less. Effectiveness of therapeutic antibodies high for the original version, and then of course high for the UK variant, but reduce and ineffective for South African one, and reduce or ineffective for the Brazilian one as well. See, there are evidence for reduced antibody protection against SARS-CoV-2 variants. Cell culture studies show that mutated viruses are less susceptible to inhibition by antibodies from recovered or vaccinated individuals. Yeah, that can be a person who has got the disease and recovered. From the recovered, then the person has antibodies, or vaccinated people have antibodies. So for both these cohorts, the protection is less for the stronger variants. See, these viruses have mutations in the so-called spike protein. This spike protein, just like these threads, they are the spike proteins, the structure on the surface of the virus that is responsible for the attachment to host cells. So they mutate so that they chain these spike proteins to make better sort of catching to the host cells. At the same time, the spike proteins also the major target of the immune response. So in immune response also the major target is spike protein. So antibodies generated in response to SARS-CoV-2 infection or vaccination bind to spike protein, thereby blocking the virus. The SARS-CoV-2 variants, this one, and are no longer inhibited by an antibody used for COVID-19 therapy. So there are various vaccinations, but these variants are no longer inhibited by. I mean, these variants are very strong, so that difficult to be inhibited by the vaccines, as this paper reports. Very high index journal paper. So there are different types of vaccines, of course, as we all know. There are different types of vaccines, and RNA vaccines work by introducing an mRNA sequence which tells cells what to build to the system, which is coded for a specific antigen. So this is RNA. Or DNA, there are vaccines, but not for SARS-CoV-2, but DNA-based vaccines. And viral vector vaccines use live viruses to carry DNA into human cells. Virus-like particles. This type of vaccine contains molecules that mimic the virus but are not infectious and therefore not a danger. Virus-like particle has been an effective way of creating vaccines against diseases such as HPV, hepatitis, and malaria. Protein subunit. This kind of vaccine uses part of the virus. This is, in this case, the protein component. It uses a protein component. These vaccines can also be used on almost everyone, including people with weakened immune systems and long-term health problems. Then you have inactivate the virus itself. We can inactivate and use it, dead version of the virus. So mRNA vaccine, SARS-CoV-2 virus. So we have spike proteins. mRNA is made with instruction to make viral proteins. So these in the nanoscale, these viral particles are nanoparticles. Nanoparticle is a particle with at least one dimension in the nano range. Nano range is 1 nanometer to 100 nanometer range. So these are actually most of the viruses are within that nanoscale, but this coronaviruses with crowns are larger than that. Bear virus is like 125 nanometers. Usually these viruses come with aerosol particles coming from your nose or mouth of an infected person. Then the size of this aerosol particle is like 300 nanometers. So usually this spike protein with this virus, we have mRNA is made with instructions to make viral protein. So this mRNA, you give instructions to make viral proteins, is packaged in a lipid nanoparticle. You see, even in this mRNA vaccines, nanoparticles are there, lipid nanoparticles. So these lipid nanoparticles make sure that these virus particles are embedded in these nanoparticles, not destroyed within the systemic circulation, that with the blood, but it goes to right places and do its action, does its action. So these are actually micelles of lipids. So lipid nanoparticles are used to encapsulate this mRNA vaccines. So vaccine is delivered as an injection. So mRNA is released into the host cell. mRNA is used to make viral proteins. So mRNA of the virus is used to make viral proteins inside. So thereby antibodies are developed and give immune response. And if you're interested in more, this is a very good article in CBC News. We can save this one and you can go to this site and read more about it. As we know, because of this pandemic situation, most of these vaccines were approved on emergency or pandemic situation. So classical vaccination phases, like the usual vaccination for other viral infections, like involves preclinical stage of vaccination about 18 to 30 months. And then phase 1 trial, dozens of clinical trials, more than 30 months. Then phase 2, hundreds of clinical trials, more than 32 months. Phase 3, thousands of clinical trials with people, more than 30 months. Phase 4, approval manufacture from 12 to 24 months. If you add up all this, it goes over several years. But in this pandemic situation, you can't follow the standard procedure. So what the approving agencies did was they approved this virus, these vaccinations on emergency basis or pandemic under pandemic situation. So now you see the stages, lengths of these stages have been drastically reduced. Preclinical stage of vaccination, no period. Phase 1, dozens of clinical trials, about 6 months. Phase 2, hundreds of clinical trials, about 6 months. Phase 3, thousands of clinical trials, no period actually. Approval in 6 months or so. So in comparison to the standard procedure that involves several years, more than 5, sometimes 15 years or so. Here it is within less than 1 year, everything is done. Therefore, there are some risks associated with this fast-track vaccine evaluation, as it is highlighted in this www.nature.com article, Nature article. The administration of a new vaccine must always be carefully associated with a rigorous study of its safety. That is very important. The safety of the vaccine is very, very important. Right? This is particularly important because a vaccine is not a drug for 6 people. So usually when you are sick, we take drugs, but vaccine is not a drug for 6 people. It is given to the healthy people. 6 people at risk of dying, but rather a treatment that is given to those who are well to prevent the risk of falling in death. So vaccine is given to healthy people to prevent the risk of getting that infected. So the race to develop COVID-19 vaccine is not only justified but necessary. But it is, it's important because the number of deaths, as you saw, is in millions. However, the time required to evaluate the dangers and risks that may arise. So, why you need such a long time of phase 1, phase 2, and phase 3 trials is that, you have to evaluate not only the efficacy, but also the side effects, cytotoxicity, what you call dangers and risks that may arise from a new vaccine. So it must be included in its development. In some cases, vaccines prepared against other coronavirus or other viruses have worsened the disease. In some cases, not for this one actually, as this paper tells us, for some other coronaviruses, the vaccines have actually worsened the case. This is induced T helper 2 type immunopathology. Claiming to have won the race to develop COVID-19 vaccine or the distribution of candidate vaccine to clusters of the population before all data from clinical trials are obtained, before obtaining all these clinical trials and carefully analyzing them can be dangerous and erode trust in both the vaccine and regulatory bodies. Actually, this is not a good thing, but it is a must because of the pandemic. But it is associated, it can be associated with various consequences, adverse consequences. See, so then, like recently, people are developing new variants or new types of vaccines also. This is one of those recently developed ultra-potent COVID-19 vaccine designed by computer. Nowadays, actually, drug design. Those days, of course, you remember in our organic research laboratory, people go for these plants, extracts, and then various trials. Nowadays, drug design doesn't follow all these steps. Instead, the disease, the mechanism by which drug is interacting with the cell or whatever the molecule. So then they use computational analysis to develop the right thing to block or damage, break or damage the culprit. So based on computational analysis, actually preclinical data published in Cell. Cell is a very high standard journal, show the self-assembling protein nanoparticle. This self-assembling. These self-assembly means these nanoparticles assemble themselves. It is self-assembly is a very old thing actually. All your organs are made of this self-assembly. All your organs are made because no one has made your ear to have this shape or your nose to have this shape. So when the right molecules under right conditions come together, they assemble to give right structures. So likewise, if you know, just like you are playing with these cards or assembling this any other thing, they self-assembly. These molecules, under right conditions and right interaction, they assemble themselves in the way to make cells, organs, etc. The nanoparticles does it. So preclinical data published in the Cell show the self-assembling protein nanoparticle vaccine spurs extremely high level of protective antibodies in animal models. So extremely high level of antibodies in animal models. If you do small amount, it produces large number of antibodies. So now people are trying to develop more and more novel type of vaccines with less and less dangers. And so if you go to this YouTube I will quickly show you how they do it.
No, welcome to the
this is a very good keynote speech by one of our expatriate scientists called Professor Sunil Vimalavans, sir. So this is a lengthy presentation actually.
We cannot see. Sorry, we cannot see. I think you have to share the YouTube screen.
Presentation.
Yeah, we cannot see it.
No, I'm not going to actually do it.
And then move to the biology of vitamin.
So I had to share again with YouTube, yeah?
Of course, you have to share the YouTube, otherwise you cannot see.
Now the thing is that this is a very lengthy presentation. I'm not going to show you the entire presentation. I'll give you this YouTube address. And please go through it. This is like more than 1 hour presentation on how vitamin C is effective in increasing your immunity. So you better go through this YouTube presentation of Professor Vimalavans. It's more than 1 hour, actually 1.5 hours or so. And it's a keynote speech done in he's a very good friend of mine living in USA. And Professor Vimalavans is a distinguished endocrinologist. He knows about these diseases very well. So please go through that YouTube one. But if I am going to show you, the entire presentation will be on that. So since we have no time for that, I'll give you this YouTube link so that you can see it. We talked about, and then we were talking about how can nanotechnology help to combat COVID-19 opportunities and urgent needs. As you can see, the nanotechnology is the technology, very simple technology, that manipulates materials in the nanoscale. Nanoscale means at least in one dimension, the size range is from 1 nanometer to 100 nanometers. In other 2 dimensions, it can be within that range, or it can be larger than 100 nanometers. So nanoparticles, nanotechnology can be used to make like autoclean. We can have nanoparticle sprays, which will naturally, when you spray onto surfaces, they'll naturally clean the viruses, bacteria, etc. And then as we have done, we can use nanotechnology to make face masks that can destroy SARS-CoV-2 and other bacteria and viruses. And we can make nanotechnically modified clothes with virucidal activity, virus killing activity, so that if the virus incidence on the cloth, it will be destroyed. The nanotechnology can be used for the detection of virus detection, sensors. And nanotechnologically modified vaccines are there. Nanotechnology-based vaccines are there now. People are developing now, still in the developing stage. And even those, the standard mRNA vaccines use nanotechnology like lipid nanoparticles to encapsulate this mRNA. And then inhibition of viral infection. Nanotechnology can be used straight away to inhibit the viral infection. And also nanotechnology can be used in many other ways, like improve the immune system response, and also surface decontamination. And so you see in the protection as well as cure, for all these purposes, nanotechnology can be used. So this is one of those strategies using nanotechnology for virus detection and disease diagnosis. So what is shown here is a schematic diagram of COVID-19 FET sensor operation procedure. Graphene as a sensimimeter. Graphene, as you know, the single layer of graphite. Discoverers have won the Nobel Prize also. Graphene is the material with highest thermal conductivity and highest electronic conductivity. Graphene is a one atomic layer thick graphite sheet. Graphite is large number of billions of those sheets stuck together just like a carpet. Right, so the graphene as a sensitive material is selected, graphene, and SARS-CoV-2 spike antibody is conjugated onto graphene sheet via this compound. So there's a compound here. The SARS-CoV-2 virus is there. You can attach that onto graphene using this simple chemical material, pyrene butyric acid N-hydroxysuccinimide ester. That is that compound. So this is the linker that can link the virus onto the graphene sheet via this material, which is an interfacing molecule as a probe link. And then you can detect this virus when virus comes. So the properties of graphene, when it's attached onto that, when the virus is attached, changes. That change you can measure. So and thereby you can correlate the change to the even number of virus particles, virions. That is one example where nanotechnology can help to detect the virus. And of course, nanotechnology can be used to make personal protection equipment, not only face masks, all these things that medical personnel who are dealing with patients require these things. So you can use nanoparticles, nanofibers. I show you what we have done at towards the end of this presentation. And nano gaps, all these things can be used to make various functions like hydrophobicity, resistance, filtration, breathability, antimicrobial properties. All these properties can be introduced to all these, like the cap, these face masks, face shield, PPT equipment, and then the smart shoes, even all these things can be modified with nanotechnology to give you maximum protection, particularly suitable for the medical person who are dealing constantly with the patients. And then, of course, this is another one, coronavirus COVID-19 nanotechnology. See, you can see you can virus detection, vaccines, virus control, nano-based personal protection. All these things are there. So, since time is running, actually, I will go into next one. Right. So, this is an interesting article where what people have done here is actually binding H-cow and this virus to host cell. So using this molecule connected with boron double OH2 and this part, you can connect this and inhibit the virus replication. So nanoparticles, these carbon content dots modified with this material can be used to inhibit the viral replication. That means as a drug can be used. So what nanometers do is nanometers also avoid viral contamination by air, and just like the face mask, and contact with infected surfaces may be especially helpful in sterilizing protective equipment and in a hospital environment. Additionally, nanometers exhibit electrical and optical properties that have been widely explored So nanometers have special electrical and optical properties, like silver nanoparticles have what is called surface Plasmon resonance effect. So those things can be used to make a point of care diagnosis kits. The sensitivity of detection could be improved by using nanoparticles, which enable us to identify the analysis of low concentration. Even low concentration of viruses can be determined using nanoparticles. These strategies contribute to rapid diagnosis and isolation and may facilitate treatment of people infected with SARS-CoV-2. So you see, it can be used to protect as well as to cure. The interactions between nanometers and biological interfaces are the basis for their potential biomedical application. Nanometers interact very nicely with biological interfaces, because biological interfaces are mostly like in the nano range. So they like each other, they interact with each other, and therefore you can use nanotechnology to not only to detect but also to protect and to cure these viral infective diseases. For example, metal and metal oxide nanoparticles are the very common materials used. I'm not going to read all these things, just to show you that these particles have very inherent properties. Using them, you can develop various things like inhibition of receptor binding. So these nanoparticles inhibit the spike proteins binding onto your cells. And they can generate reactive oxygen species to destroy these viruses. They can degrade this viral envelope. They can, I'll show you in our research, they can break down these things and destroy this whole structure. The glycoproteins can be denatured. So in various ways, these nanoparticles can destroy these viruses. Silver nanoparticles are one of those common materials, but the trouble is silver nanoparticles have some cytotoxicity effect to healthy cells. Like we need to use very small concentration, like 1 to 10 ppm, but above that, like concentration about 20 parts per million and above are toxic. So therefore, we have to be careful when using silver nanoparticles. That is why we never use silver nanoparticles in our face mask materials. And we have used actually, we have determined the antiviral activity. This is our research now. Titanium dioxide, copper oxide, and zinc oxide nanoparticles against respiratory syncytial virus, RSV. This is, in Sri Lanka, we have to have actually very third level 3 safety cabinets, safety rooms to work with SARS-CoV-2, but in collaboration with Professor Fasiha Noordin, she is the virologist in our microbiology department of our medical faculty. We evaluated the antiviral activity against this similar virus called respiratory syncytial virus, RSV. So we see that titanium dioxide nanoparticle concentration percentage 0.5. Without that, and RSV antigen cells per field, 30 survived out of 50. So as you increase the concentration to like 0.1 to 1 survive. Again, of course, the results are not actually proportional. You see, as you increase the concentration, the number is again increasing. So we use actually this copper oxide. She has given copper. Copper oxide also, like we see with even very small concentration, 0.06, like 2 out of 50 survived. 0.1 to 1 out of 50 survived. So with very small concentration, you can kill the virus or destroy the virus, but trouble is copper oxide is also toxic. Although in America also now copper oxide based face masks are available, copper oxide is not very friendly for us. So although we have determined, we never use copper oxide also. And zinc oxide is a material that we have used. The nanoparticles, as you can see, without zinc oxide, with time also, the this the virus destroys, so 30 out of 50 remains. But within that time, if we use like 3%, but if we use half the dosage, 1.5%, nothing remains. Even the quarter of dosage, nothing remains. 0.37, that means 1/8 the dosage, only 1 out of 50 remains. So fields, not only the viruses, virus fields we are talking about, large number of viruses in one field. So you see that zinc oxide nanoparticles are highly efficient, very efficient to destroy this RSV virus. So those days, because we did not have facilities to do this actually SARS-CoV-2 test, we worked with bacteria and this RSV virus. But then, of course, this test can be done from London Virological Research Services, which is a part of Imperial College. And because I'm an alumnus of Imperial College, I mean, they agreed to give me at a lower rate, like it's still 5,500 pounds a test. That is like 1.7 million rupees. So nevertheless, we did it. Otherwise, it's 13,000 rupees. 13,000 pounds actually. So we did that with SARS-CoV-2 also. So this is how these tests were done. I think I don't want to bore you with all these procedures, but this is how our experts in virology did the RSV test using fluorescence microscopic visualization. And they confirm that dose-dependent activity is obvious with zinc oxide only, not with others. So we use zinc oxide nanoparticles as viricidal materials. But the nanoparticles that we use were like not spherical, but the core is spherical, just like the virus. We have this star shape. The sharp blades coming out of this sphere in the middle. So in all directions, these blades in the nanoscale are very, very sharp, so that they can basically damage the spike proteins, cut the envelope, and destroy that virus into pieces, as I will show you later. So that is the major action. So most of these the microbes can develop resistance to chemical agents. However, for this mechanical action, just like you're cutting the parts and bits and pieces of the virus using scissors, these sharp blades of nanospheres can destroy and break and cut into pieces. So that is how they destroy virus and bacteria. So we find that zinc oxide is very effective in doing that. So we did with various bacteria. I mean, actually, we have done these experiments for several years. I have produced 3 or 4 PhDs in zinc oxide-based textiles, antimicrobial textiles, starting from Dr. Srimala Pereira. She is the head of Department of Textile Technology Department of University of Moratu, actually. So we see large inhibition zones with this bacteria do not grow. Even if that is because, I mean, these nanoparticle surfaces are positively charged. Viral envelope or bacterial envelope is usually negatively charged. So therefore, the electrostatically they attract not only on the surface but also in the vicinity. So that attraction, what we call, I mean, that is called the nurse diffusion layer, has a limit. So within that range, the bacteria cannot survive because it comes to the cloth and modified cloth and destroys. So with 3% zinc oxide, so they have tried 3% zinc oxide with 1% copper oxide, etc. With control, there is no that. So with cloth 3 zinc oxide 3%, small, I mean, doesn't bacteria on the cloth all dead, and also in the vicinity also a small circle bacteria. When you add more, at least 1% copper oxide, the cumulative effect, synergistic effect makes it even more strongly attracting, so that the inhibition zone becomes larger. So tried with 3 different, like gram positive, gram negative, and also very dangerous bacteria like Pseudomonas. And then actually our mass we got tested from Bureau Veritas actually we have to do 13 tests. Bacterial filtration efficiency. When you tested that with, actually, if you do that particle filtration with Slintech, you don't get the accurate values, because in Slintech machine, what they do is not correct actually. The particles in the air is sucked through the mask and then analyzed. But it depends on how much particles in the air. So usually polluted air should have at least 10 to the 20 particles. So it is not a good thing, but it also measures only the neutral particles. But since bacteria viruses are charged particles, so you have to use bacterial filtration efficiency. So which we did from ITI, and we got 99.99% bacteria filtration through our modified cloth. I'll show you. Then splash resistance. So at a particular pressure, you splash blood onto the mask material or the mask and then measure whether blood penetrates through it. So that is important for surgeons. So the rate is like higher rate than the blood coming from the cut arteries. And then also differential pressure. That is when you have a mask, there is a pressure difference. Pressure is created inside. So without mask, there is no pressure difference. So the pressure difference between your face and the mask in the head space is called the differential pressure, which is measured for good breathability. This differential pressure should be very, very low. Imagine it's 4 water millimeter per square centimeter. So our mask pass that also. And then dimension of the mask. So with certain dimensions are there, it passes. Dimension on the nose piece also dimension pass. Then number of pleats and layers in the mask pass. Safety is a microbiological cleanliness pass. Flammability. So this, because our masks are made of cotton materials, unlike this polypropylene materials, so the cotton takes time to ignite. So usually if you are wearing a mask and if there's sudden fire is there, so we need to give time to remove the mask. So at least 10 seconds. So usually these, I don't want to mention names, but polypropylene type masks that are used have like less than 10, this is 10 second, that minimum one. However, this cotton is not that flammable, so therefore it takes about 20, 30 or even 1 minute to burn. So that means we are giving enough time. So flammability is best passed with this cotton mask, and pH and all these parameters pass. And also there are ample literature in ACS Omega and various other standard journals on how zinc oxide nanoparticles can destroy SARS-CoV-2. And then, of course, our masks are designed to make them usable for repeated washing. One mask can be used at least 25 times washable in standard washing machines. So within we calculate zinc oxide ppm, hardly changes. You see 10,599, almost 10,000 similar range. Even if you wash for it, that is because these nanoparticles are chemically bound. So that we did this research on particularly on mask materials for more than 1 year to make sure that they don't detach. And then we pass an airflow at a rate much higher than your breathing rate. Breathing rate is 6 liters per minute, but we pass airflow at 10 liters per minute or higher for 24 continuous hours, and measured both the titanium dioxide microparticle and zinc oxide nanoparticle amounts. There's no change whatsoever. And then percentage reduction of bacterial count against control sample 0, but this is 99.89, 99.32. So almost higher than 99 point bacterial count reduction, even after 25 times wash samples. So many people are coming in the waiting room now. And also the safety. So that was an issue that the microbiologist brought for several more than several months to get a NMR certificate. I mean, we know that they are non-toxic, but then we had to do this toxicity and hypersensitivity test using mouse models, and we had to sacrifice like 50 mice, unfortunately, but not me. The Professor Jayanta Rajapaksa, professor of veterinary pathobiology in our vet faculty, did it. Our product name Respirone Nano EV99 face mask, and over 3 months. So then what he did was he had a control mice sample, and then the test mice sample. For the control mice sample, the normal air comes. For the test mice samples, air always comes through the mask. They don't get any normal air coming from the mask. It's actually normal air, but it's coming through the mask. And then just before treatment, and 3 days after, 15 days after, 30 days after, 6 days, and 9, over 3 months measured the normal air. There's hardly any change. This is the weights of the air through mask. The mice subject to air through mask, the weight doesn't change. And also measured the histopathology of lungs, kidneys, livers of normal air exposure and air exposure through mask after 3 days, 15 days, etc. So then you see that there's no control 3 days, up to like 3 months. That is for mice, 3 months is more than enough. So there's no change whatsoever. So that means that is because, I mean, we knew that because our nanoparticles, our materials used in these fabrics are chemically bound and are not detached due to airflow. So there's no chance for you to inhale them. So we have done this test for 3 months, and we have this big report, part of that I have put there. And the conclusion: there are no any pathological lesions observed, either hypersensitivity or diffusion of collagen, and these are the things that can deposit if a particle goes. Proteoglycans remolding the extracellular membrane of underlying epithelium of the air passage. Therefore, there is no any toxicity effect to lungs, liver, and kidney tissues. Based on the above study, the breathing through Respirone Nano EV99 face mask has no detectable adverse effect in acute or chronic exposure in the respiratory tract or the lungs, I don't know what is Nanometers tissues. Dr. Rajapaksa is a chair and senior professor of parasitology. So then it's great. So then actually these microbiologists were against this one, but then they could not go for that because we have the report saying that it is not toxic at all. We knew that, we have proven that, but they saying that particles do not detach, but they demanded this. Actually, we did it. And also, I mean, we can measure titanium dioxide and zinc oxide contents using XRD before wash, after several 25 washes. You see the amounts measured using XRD hardly changes. See, hardly changes. So this is what I said: before air passing, after air passing for at high rate for 24 hours, there is hardly any change. So 45.6, 19.2, 45.20, almost the same ratio. That shows. So this is a video that you can enjoy about our face mask, and I'll brief you on what they are.
Respirone AV99 is an environmental friendly, biodegradable, low cost, reusable and antimicrobial face mask with water and blood repellency and stain resistance. It also repels and destroys any aerosolized viruses or bacteria. In this multifunctional face mask, the target has been to reduce both the waste burden and the impact on the environment. As such, Cotton, a commonly available, natural degradable fabric material, has been used in 3 layers, nanotechnologically modified individually, to have unique, inherent properties. The outer layer cotton fabric is super hydrophobic to repel any aerosolized viruses and microbes as well as blood or any other water-based stains, comparable to a lotus leaf shedding water. Any microbes penetrating the outer layer will reach the nanotechnologically modified middle layer with multimodal antimicrobial activity. Micro and nanoparticles are used to partially block the pores of the woven cotton fabric via chemical bonding to a level of 300 nanometers, resulting in nanometric range filtration. The adjustable ear loops ensure tight fitting to the face of the wearer. A unique mask design and a metal or plastic noseplate addresses the problem of air escape at the nasal bridge. The innermost layer is made up of 100% hydrophilic cotton to absorb carbon dioxide and moisture present in the exhaled air. This face mask can be reused even after 25 cycles of washing or disinfection, at least for a period of 1 month. The nanoparticles used in these masks are safe in a wide range of applications, including medical and cosmetic industry. High airflow tests and wash cycles have proven that the chemically bonded nanoparticles are stable and do not detach from the fabric surfaces. The mask has medical-grade characteristics with high resistance to flammability, satisfactory results in differential pressure tests for breathability, and is highly resistant to blood and fluid permeation.
So this is the team, myself, Dr. Chamindera, the consultant radiologist, and my student. So I will quickly go through this is the outer layer. You see that if your blood comes, it stays as a sphere. When you tilt it, it falls off, just like the water on the lotus leaf. So this is with the superhydrophobicity. The dislikeness for water is measured by measuring the contact angle. So if the contact angle is above 150, we call the material is superhydrophobic. If it is above 90, a contact angle, it's still hydrophobic. Below 90, it likes water, hydrophilic. So you see, at least up to 20, you see is superhydrophobic, and up to 25, even 30 is hydrophobic. So it's so therefore we recommend, because this is the thing, because due to wear and tear always it's possible to physically damage. If physical damages when you wash so many times, you see contact angle remains above 150 or 20 washing cycles, and even 30 washing cycles you can go still hydrophobic. So after that we don't recommend, because this property gradually decreases. But other properties down, so at least 25 washing cycles you can go. So here that's how we, cotton, when it's a woven fabric, we have yarns, say horizontal and vertical. Between them you have pores. So we use a very high sensitive light microscope, and you see that the cotton surface has pores. As you block these pores, gets reduced. As you block completely, the light cannot go through at all. So there are that means that pores are reduced down to at least the smallest number of visible wavelength, that is 400 nanometers light. Actually, when you do at SCA, you see that you have reduced down to 300 nanometers. That we did by blocking with right size. These are like 39 micrometer pores, so microparticles of titanium dioxide. The surface of which is positively charged, so that if virus comes to mid layer, then it is electrostatically bound and destroyed. And also the pores do not let any aerosolized particles to mechanically go through also. So you see that with different extent of coverage, you see different extents. At 0.3 micrometer, that 300 nanometer is the size of the aerosol particle. So when you measure from slim tech, still you get 67 plus 4 with that. So as I said, slim tech measurements are not reliable. Above that, you get 91%. But this, even if you do N95, you don't get 95 is slim tech because the method itself is strong. But when you do bacteria filtration, we see that with that, we get 99.99% bacteria filtration efficiency. And this is RSV antigen test results. These are bacteria results. Even with Pseudomonas, is a very dangerous bacteria. With 3%, you can kill all of those bacteria. This is ITI test report. Bacterial filtration sample on 99.8 sample 2 99.99, 99.99, 8 not 99 like that. So 8 not to 99.99% bacteria will make sure that these size particles do not go through this middle layer. And the inner layer is a soft inner layer with excellent moisture absorption. So actually, this is demonstrated like this. You put some dye, it immediately absorbs into that. So in the perspiration and in the exhaled air, you have carbon dioxide. So you want to get rid of that carbon dioxide. You should not let carbon dioxide accumulate between your face and the mask. It's the head space. In some masks, of course, this carbon dioxide accumulation is there. That is why you have one-way valve. So the one-way valve in those masks, but that is banned because if you are a patient, then of course, I mean, all the viruses, the aerosol particles immediately go into the air with the one-way valve that is open out to the atmosphere, towards the atmosphere. So that is banned. In our case, it is quickly absorbed because cotton, it likes water, it absorbs and quickly removes from the headspace. So there's no, because I'm actually jogging for 1 hour, like 1.2 kilometer path, 6 rounds at least, and with wearing this mask. I don't get any problem because of the breathability is adjusted to the right level, and also this carbon dioxide and water escape is also there because it's cotton. So inner layer with excellent moisture absorption, wicking and evaporation, free of fluid clogging, and non-accumulation of carbon dioxide and no re-inhalation. Some of these masks which have poor carbon dioxide absorption gives the carbon dioxide rich air in front of your face, in the headspace with the mask and the face. And then because carbon dioxide is heavier, when you inhale, what you are inhaling is always carbon dioxide rich air, so oxygen content not enough. There were cases where the mask wearers were admitted to ICU even. So you have to be careful. So the material like these things, we have taken all these things to make sure that you have no problem of carbon dioxide accumulation and re-inhalation. It's a barrier between phase airway and filtration layer. So therefore, even if they are not the material that used to modify in the inner and the outer layers, so this is a barrier. So whatever the material, they are the materials that are used in face creams also, but we make sure that they are not even touching your face. There's a barrier. This is a barrier. Unmodified super hydrophilic cotton barrier. So comfortable to face because it's cotton, safe and non-allergenic. So we have tested with 500 people, randomly selected, and there were no complaints of allergic reactions or any things. We have given a questionnaire with several questions to answer yes or no. So with 500 people, and its ease of wearing, all these properties are there.